Hi everyone, in today's video I'm going to go through non-disjunction in meiosis I and II. So what is non-disjunction to start with? Non-disjunction is a process that occurs in anaphase of either meiosis I or II and essentially can result in an aneuploidy. An aneuploidy is when the amount of genetic material in a zygote or even in a haploid cell is not at the normal amount. So let's start off. I think this is best represented diagrammatically, so that's why I'll draw it out first. So if we look at non-disjunction in meiosis I, we start off with the parent cell. And this parent cell is what will form eventually the 4 haploid cells or 4 haploid daughter cells. So this parent cell contains its own genetic material, which, and we'll skip over the stages of meiosis just for this video, but we'll start it off in the metaphase stage. So, as we know from mitosis and meiosis, in the metaphase stage, the chromosomes of the cell are aligned along the metaphase plate, which is at the equator of the cell. And these two chromosomes are aligned so that they can be pulled apart by the centriole pairs and the kinetochore microtubules that extend from these centriole pairs, as so. So what essentially happens in non-disjunction is that the chromosomes aren't able to properly separate from one another. And we know that in meiosis I, the chromosomes are actually in the form of tetrads. And tetrads are basically two homologous chromosomes that are joined together by what are called cohesin proteins. These proteins hold the chromosomes together and ensure that they don't undergo this non-disjunction. But as we know, that mutations can occur in some cases. So, say for example, non-disjunction happens in meiosis I. So after metaphase, which I'll abbreviate to M phase, and in anaphase, which is I'll call A phase, one of the chromosome pairs will essentially stick to one another. So in terms of this chromosome pair that's at the top, let's say that separates properly. There's no problem there. All right. So what happens here is that the two homologous chromosomes are able to split, no problem, all good. But for these bottom chromosomes, say for example, there's some sort of malfunction in the chiasma, which is or chiasmata, which is the points at which the two chromosomes cross over one another to introduce genetic variability. Or say, for example, the cohesin proteins aren't working properly. Say that they stay stuck together. And as we see in this picture here, there are two chromosomes present on this side and no chromosomes present on that side. So already we have a problem because normally there's supposed to be one chromosome present on each pole of the cell. So what happens after that? So if non-disjunction occurs in meiosis I, after meiosis I, we have, of course, two cells forming. So because two homologous chromosomes went to one side, one of these cells will have three three chromosomes, and one will just have one because we have this chromosome at the top in addition to these two chromosomes at the bottom going into this cell and just this chromosome going into this cell because there's no other chromosome present. Now, after that, what happens is quite interesting because unfortunately, there's not any checking or particular proofreading mechanism that can correct this after it has occurred. So, in meiosis II, the extra two haploid cells that are formed essentially just take on the mutated chromosome structure that the two previous cells had. So, during meiosis II, let's say that these chromosomes once again align along the metaphase plate, 3 in one and 1 on the other. And what will happen in anaphase is that in the formation of the 4 haploid cells from this one parent cell, right, there will be 3 chromosomes present in 2 of them and only 1 chromosome present in 2 of them, because these 2 chromosomes split apart as from the centriole pairs, and these 3 chromosomes also split apart. So what does that mean? And just for reference, I'll draw a normal haploid cell, which has 2. So we call this cell in having an N conformation, because N is what denotes having 23 chromosomes. So because this cell has 3, it will be N plus 1. This cell will also be N plus 1. This cell, because it only has 1 chromosome present, it will be N minus 1. And this one will be N minus 1 as well. And we have special names for these particular cells. Because this cell has 3, we call it a triploidy. Trisomy, sorry. Because we have 1 extra. And because these cells have 1 less, we call them a monosomy. And trisomy might ring a bell for you, because Down syndrome is actually called trisomy 21 because it has 3 or 1 extra copy of the 21st chromosome. And that's what non-disjunction in meiosis I is. Now, in terms of non-disjunction in meiosis II, overall, the process is quite similar. So what happens in this stage is that originally we have this parent cell with its chromosomes aligned along the metaphase plate, 2 centriole pairs with their microtubules attached. The only difference between non-disjunction in meiosis I and meiosis II is that the genetic mutation occurs in meiosis II, as the name suggests. So in the formation of the 2 daughter cells in meiosis I, everything happens normally. So this means that these chromosomes will separate properly, essentially. So we have 4 chromosomes present on this cell and 4 chromatids present in this cell. Now, the mutation occurs in meiosis II, which means that if we align these chromosomes along the metaphase plate, they will not separate properly. And non-disjunction in meiosis II usually occurs only in one of the 2 cells. So what that means is that unlike meiosis I, we will have 2 cells that are normal. So unlike meiosis I, where there are 2 trisomies and 2 monosomies, in meiosis II, there will be 2 normal haploid cells forming. So say, for example, we chose this cell that undergoes non-disjunction. So in terms of this cell, 2 haploid cells will form properly. And say, for example, this chromosome doesn't separate properly. So what will happen is that because this centriole pair will pull this chromosome to that side and also pull this chromosome to that side, whilst this centriole pair will pull this chromosome to that side, this cell will have 3 chromosomes present. This cell will only have 1. So the outcome of meiosis II is that 4 haploid cells are formed. However, there's one trisomy, one monosomy, and two normal haploid cells forming. So in summary, non-disjunction in meiosis I is when the chromatids, sister chromatids, are unable to separate properly in anaphase I. That essentially means that there is an unbalanced nature of chromosomes further on in the process and results in two trisomies and two monosomies. So there is essentially a 0% chance of there being a normal genetic structure being formed. However, in meiosis II, there are no problems during meiosis I. So anaphase I undergoes its process properly, but the problem occurs in anaphase II, where the chromosomes of the two daughter cells that had formed from meiosis I are unable to separate properly. And that means that one of the cells will form a trisomy and a monosomy, and one of the cells, which usually has a normal process having undergone, it forms two haploid cells. So there is still a 50% chance that the growing fetus will have a normal genetic makeup.